Yeah, I just wanted to briefly say we have this awesome seminar today and quite, a lineup for the rest of the year coming up. But if you're interested in presenting, please speak up because we have more spots towards in the fall part of the year that we can fill up if people would like to present later on in the year. Hugo is going to give a talk next month on his experience over the last couple decades in the field. So that is one you guys should go to. And I think we have another one lined up in July, right, Jason?
I think so. Yes, I think you may be hearing about neuroblocks in July.
Yeah, so plus a handful of others. So yeah, but please let us know if you guys want to present, and we'll get you on the schedule. So yeah.
Yeah. And also, if you have any other ideas for the working group, please feel free to email them. And we've discussed maybe having kind of a more kind of a networking or social kind of activity at some point where we can get to know each other more. Um, uh,
Yeah, I think
yeah.
a career networking event would be really helpful too, where we don't spend the whole hour presenting science and have more time for people to interact with each other. But yeah, should we maybe go ahead? So, or do you want to wait a
Yes.
few more minutes, Jason?
Um, I think we can. I'm just admitting people. I will keep watching the waiting room. I think in the future, this is our first time using Zoom today, so thank you for. Hopefully, there are no issues. Nice thing about Zoom is we can record more easily, and I may turn off the waiting room in the future because I think that's might make it a little more
Oh yeah, that
streamlined.
that's good. Yeah, that's a good idea. But the key is we can record and then hopefully post so people that are across the world can appreciate and listen to some of the talks that we have here. So if it's
Mhm.
not the ideal time for them.
Yeah. So yeah, I think with that, I can maybe go ahead and introduce our speaker today. So we're very pleased and happy to have Dr. Vignan Muddapu joining us from India. It's much later there, so we really appreciate his working with our schedule today. He's an assistant professor at Azim Premji University, where he's building a lab. His research spans computational neuroscience, multiscale modeling, systems biology, and neuropharmacology, with his focus on Parkinson's disease and basal ganglia circuitry. Previously, he was a simulation scientist at Blue Brain Project and completed his PhD in computational neuroscience at IIT Madras and was modeling basal ganglia and neurodegenerative disease there. I think his work is especially relevant to NeuroQSP because it connects molecular and cellular mechanisms with circuit function, levodopa response, motor behavior. It's really nice. He makes his models open source so we can all benefit from it. If anyone joined my presentation a few months ago, I borrowed certain elements from his model. And so he links pathways with circuit function, and then I combine that with kind of the hypothetical gene therapy and MOA. So we're very happy and delighted to have Dr. Muddapu here today, and I will hand it to him for his presentation. Thank you.
Yeah, I'll just share my screen. I'm not allowed to share the screen.
Oh, okay, working out the kinks. Hmm. Does it? Oh, okay. Can you share now?
Yes. Are you able to see the screen?
Yes, thank you.
Push this out. Yeah. So hi all. I don't think here it's night. I don't know what is the time there. It's morning, I guess. So I'll just start away with the presentation of what I've been doing from last 10 years. So the title is just the title of my paper, which was published 3 years back, and what we have done beyond that. So that will be the whole presentation is about. And to start off with, here we developed a cortical basal ganglia model, where using this model, we try to perform a task where here there is a goal and it arms tries to reach the target from certain initial condition. And how basal ganglia on cortical loops helps in achieving this. So, and we developed at different scales in terms of time scales, temporal and spatial, both. And we studied in normal condition, in Parkinsonian, and also in the medication condition. Here it is Levodopa. So before going to the details of the model, we'll just understand what are the brain regions involved in this modeling effort. Is the basal ganglia, which is the core, heart of the whole modeling work, where it receives cortical inputs from different cortical regions and comes to a region called striatum, which is an input nucleus of basal ganglia, where it's a combine of caudate nucleus and pituitary. And then from there, it diverges into two different pathways. One is the direct pathway, where the output nucleus of basal ganglia, which is GPI and SNR, where the direct connection from the input to output is the direct pathway. And the indirect pathway is via GPE STR. So here I'm just talking in terms of abbreviation. I hope everyone familiar with the basal ganglia abbreviation. So I'm just not going to details of explaining the different nuclei. And from there, the output comes to thalamus and back to cortex. So the balance between this direct and indirect pathway, where activation of direct leads to initiation of movements, activation of indirect pathway leads to inhibition of movements. That's a gross understanding. And the balance between direct and indirect pathways maintained by the dopamine, which is the dopamine projections coming to the striatum from subthalamic, from substantia nigra pars compacta, which is having dopaminergic projections from these midbrain regions to this striatum. And there is also another pathway where the direct from cortex, the connections comes to STL, which is called as hyperdirect pathway. So in this modeling effort, we haven't considered the hyperdirect pathway yet. So in future work, we might consider, but as of now, we are only sticking with the direct and indirect pathway because we want to see how it is involved in movement. So these are the different functional loops of basal ganglia, which are involved in broadly three broader functions. One is the sensory motor functions. Another is association cognitive functions, and another one is limbic functions. So where the information from the cortex comes to, in the case of sensory motor, comes to dorsal lateral striatum. From there, it goes to pallidum. From there, thalamus, back to cortex. Similarly, in the case of cognitive function loop, information comes from the cortex to dorsal medial striatum. From there, again, pallidum. From there to thalamus, back to cortex. Similarly, in the case of limbic, it comes to nucleus accumbens, which is the ventral striatum. From there, it goes to, again, ventral pallidum. From there, it goes to thalamus, back to the cortex. So these are the broader functional loops of basal ganglia. And if you want to go specific, like different functions involved in terms of motor, in terms of cognitive, there are these few functions which I have listed here. One is action selection, sequence generation, goal-directed behavior, anything which involves working memory, exploratory behavior. If you want to explore, trying to learn something, how you'll explore the task, timing of the actions, reward-based learning, gating the actions, and motor preparation. So basically, involves all these functions. So here, so one of the major disorders which involve basal ganglia is the Parkinson's disease, where we already discussed, which is a normal pathway, which is the direct pathway, and this is the indirect pathway. And this balance between direct and indirect pathway is maintained by this substantia nigra pars compacta, which is the dopaminergic projections coming to striatum. But in the case of Parkinsonian, what happens? These particular projections are lost, which lead to imbalance of the direct and indirect, which lead to more of indirect pathway getting activated, which leads to inhibition of movements, which is bradykinesia. So here on the right side, you can see the cardinal symptoms of Parkinson's disease. One is the tremor, which is involuntary movements, rigidity, stiffness of muscles, bradykinesia, slowness of movements. And you can see the last GIF, which is a shuffling gait and also postural imbalance. Like they will be having stoop posture, like bending towards, and the balance will be very much affected. And in my PhD, I have done some work of why are these cells dying in the first place? What is the reason? So the hypothesis there was energy deficiency at subcellular, cellular, and system level can be a major cause of this SNC cell loss, which might lead to different cardinal symptoms of Parkinson's. I'll not go into details of this work. I'll just from the whole my PhD work, one major thing was this developing a subcellular, very detailed SNC model, which is a dopaminergic neuron model, where we use this model to do further analysis how the changes at subcellular level can affect the results in behavioral symptoms. So to study that, we focused on first is the sensory motor loop, where all taking the sensory information and how we'll respond, how we'll produce the motor function. So for that, we broadly selected the action selection, goal-directed behavior, and exploratory behavior and reward-based learning. So here the task was arm-reaching task, where it tries to reach the target. So that is where it's trying to reach the target as goal-directed behavior. And in terms of action selection, is it going in the right direction? That's the action selection. If it's going opposite direction of what the goal is, then it's need to change its action. So this particular functional feature also being studied in this particular task. And also in terms of exploited behavior, if they're not going in same direction of the target, what are the target trajectory I should follow? So it will try to explore. So based on it will learn based on the reward, it will try to learn the action. So first trial, it might take longer time. So as the trial improves, the number of trial increases, it might be faster. So all these components are involved in modeling this particular task. So this is a broader overview of the whole model. So which are brain regions involved? One is a prefrontal cortex, where your goal information is there, and your motor cortex, where you send the motor commands. You have proprioceptive cortex, where you receive the sensory inputs, and you have subcortical, which is basal ganglia, and thalamus, which involves in correcting the movements. And here we also shown the cerebellum, but as of now, we haven't included cerebellum in the model, but these are the broader scales. And we also have motor neurons in spinal cord, which performs the action. And if you go into the basal ganglia, again, we have the direct and indirect pathway. So the information cortex comes to striatum, goes out through thalamus. And the SNC, we have the detailed model of SNC, which has all subcellular mechanism which involves in this dopaminergic neuron. So this is the schematic same diagram, which proper block diagram, where again PFC has a go direction. So these are the different loops. I'll go one by one. What are these loops? First, we divide these into 3 loops. One is the outer sensory motor loop, where the information of the arm is taken by the proprioceptive cortex, and the motor command from the motor cortex goes to the motor neurons and performs the movement. And the goal information is in the PFC. So this is the sensory motor loop, outer loop. When it comes to the next loop, which is the inner loop, where the information about the arm position from the cortex comes to striatum, is the striatum does the evaluation. Is the arm going the right direction or in the opposite direction? So based on that, it will send the information to, again, motor cortex via thalamus. So this activation of direct and indirect pathway depends upon the where the error is. So this calculation error happens at the striatum. So how it's happened? The next loop, which is a central loop, where the error is calculated using this central loop, which is the nigrostriatal loop. So based on the error, the activation of that particular direction of the trajectory to which the arm is going to move is in right or not. It will calculate the error. Based on that, the reward is given to the striatum. Based on that particular set of actions will be shut down. So I'll just pause here. If you have any questions, so these are the different functions of the model. If you have any questions, I can happy to answer. Hello, guys. Anyone here? I'm the only person here. Any question, guys?
No question for the moment. Thank you.
Oh, so shall I go ahead?
Yes, please. That's fine.
So yeah, these are the broader functional different loops of the model. And then we just will go into the results. So this is the model performance in reaching task. So the blue ones are the normal condition and the green ones are the PD conditions. So you can see the time to reach the target, you can see for the control is slower compared to the PD condition. So we earlier developed a model which is cortical basal ganglia model with just the rate-coded neurons. But in the present model, we have a multiscale cortical model we have where we also have a detailed SNC model and how different this dopamine is affecting the activation of direct pathways are being involved. So that's what we're comparing with the earlier model with the present model. So I will come to what is this PD1 and PD2 later, but you can understand both are Parkinsonian conditions. So you can see in the case of our model also in the normal condition where the dopaminergic loss is not there, you can see the arm is almost reaching to the target, similar to the experiment. And in the case of other Parkinsonian condition, you can see this higher. Same with the time to peak velocity. So it will be lower in the case of normal and will be higher in the case of PD. And peak velocity, in case of normal, it will be higher. In the case of PD, it will be lower. That's what you see in the trends here, decreasing trends. And the average velocity will be higher for the normal and lower for the Parkinsonian conditions because it is very difficult to move because of the loss of this projection. So one of the predictions from our model was how the percentage of cell loss can affect this parametrics. So here you can see the different percentage of cell loss, how the movement time is increasing. You can see which 5% in cell loss, 25% cell loss, 50% cell loss, and 75% cell loss. This cell loss is happening in the case of SHC, which is the dopaminergic neurons. So when the dopaminergic neuron loss is there, how much time it takes to reach the target. So you can see how the percentage increases, the time to reach the target increases. And also you can see the time to peak velocity increases. Same with the peak velocity and also the average velocity. So from this, we can say that as the percentage of essential cell loss increases, the performance deteriorates. Means the time to reach the target will increases. So now after this, we want to understand, can we, using this model, can we actually simulate the cardinal symptoms of Parkinson? Can we, with different combinations or different parameters or different way of architecture, can we see or we can simulate these Parkinsonian cardinal symptoms? So for that, we hypothesize that if there is a differential dopaminergic projections loss, it might lead to different cardinal symptoms of PD. So for that, so you can see the top one is the normal condition where the SNC has normal projections to striatum and also to the subthalamic nucleus, where we see intensively in my earlier work where how why are the cells dying in the first place? We see that the projections from SNC to STN and STN to SNC plays an important role in their survival. So we, for our condition, we also assume that the projections to subthalamic nucleus also plays an important role in Parkinsonian compared to your projections to striatum also. So here we are considering putting the subthalamic nucleus as central because that's where you see all these Parkinsonian oscillations. So we want to see where the activation from the laterals and activation currents from the externals, how it is going to affect. So in the case of normal condition, you can see the lateral input currents is lower compared to the GP, which is one of the input currents to the STM. And you can see the in terms of the network activity, you can see that it's normal. There is no oscillations. But what happens when there is a SNC projection loss to striatum, which leads to higher equal activation of laterals and external currents to GP currents to the STM, which leads to akinetic, means rigidity type of movements. So we assume this. These are just the assumptions that we, because of this activation, different loss of these different projections might lead to different behavioral symptoms. So we assume that this might be the reason. And another one is when there is a projection loss to STM. So what will be the activation? You see the higher activation and what will be the leads to more like tremor dominant behavior, where it will where the nigrous subthalamic nucleus axons are lost. In the case of earlier one, which is the nigrous striatum. In the last case, where both the axonal projections are lost, which lead to more like a mixed behavior. So we'll see if this type of loss of different fibers from dopaminergic neurons to different nuclei in basal ganglia leads to any behavioral changes. So that's what we see in the model, where this is the normal condition. So the first figure is the distance to target. So here the target is at the distance of 0.8 meters, and the agent has to reach the target. So that's the task. And what is the second plot is the velocity. So always the movement will be bell curve. So initially it will be low, as in the middle of the trajectory it will be high, and as you reach the target, the velocity will be low. That's what you see in the normal reaching task. And this is the Asian activity. And you can see this is the two joint arm where it's trying to reach the target, which is the this circular thing.
Can I ask a question, or would you rather we
Yeah.
wait until a later point?
Sure.
So I'm trying to understand the velocity and also the reaction time measures. So first, when an action occurs, and there's a change in how long it takes for it to occur, is there an integrator that is building up a decision to take an action that is taking longer to build up, or is there a change in how quickly a signal propagates? What drives the change in the time to action?
In terms of velocity?
Yes, and I guess how is velocity calculated?
So there are different ways. So at the sensory motor loop, there is a trajectory, right? So how if you are going towards the target, you can see this normal activity. The integration happens. But at the basal ganglia also, if the trajectory is right, means the error is low. There is a race model at thalamus where it tries to race towards the target. So based on that, information goes to the motor cortex. From there, it will go to motor neurons, then to the arm.
And do you have a grid of neurons that map out the 2D space, and then the activation moves through the grid?
Yes, exactly.
And then the velocity is
this 2D space is mapped onto the PC, which is proprioceptive cortex. Again, that is mapped onto the motor cortex. From there, it goes to the striatum.
Okay, that makes sense. Yeah, thank you.
So here, this 2D space again. So here we used, in the case of proprioceptive cortex, we used a SOM, self-organized map. And in the case of motor cortex, we use a combination of SOM and AN, which is continuous attractor neural network. And the PFC is again a SOM, where we have the goal information in the 2D space again. So this all information comes to striatum, where striatum again is a 2D SOM. So can I move forward?
Yeah, that makes sense. Yeah, thank you.
Yeah. So now comes the how to simulate the bradykinesia. So we have different set of things. One is the two things we can control is the percentage of cell loss and also which axons are getting lost. Is the projections to striatum or both projections to striatum and thalamus? So to produce a bradykinesia, we need to have at least 25% cell loss in SNC, and the projections to both striatum and SNC, striatum and subthalamic nucleus should be affected. So that's why that's when you see this bradykinesia, which is slowness of movements. That's what you see. It's longer time, and the velocities you have a bimodal peaks. So this way you can simulate the bradykinesia, slowness of movement. And if you want to simulate the tremor, you need to have 70% cell loss, and it is only affecting striatum. So you can see the movement towards the target is oscillatory, and also the velocity is also up and down. So, and one interesting thing we observe is if you start again, reaching the target almost near, it is faster. But when it's about to reach, it's hangs. It will just oscillate. It's not able to reach its target. So one of the interesting things we found in this model. And the last one is the rigidity, where we see that at 70% cell loss and also both loss to both loss of axonal projections to both SNC, striatum and subthalamus nucleus leads to rigidity of movements where your muscles are stiff. You cannot, you can see the distance from the target is low and the velocity is very low. So with this, we are able to explain most like 3 of the cardinal symptoms of Parkinson disease, which is bradykinesia, tremor, and rigidity. So next will be to understand if levodopa, which is the most prominent drug given in the case of Parkinson disease, can this giving this precursor of dopamine can influence the behavior of our model. So for that, we developed a pharmacokinetic model where the levodopa dosage is given, it goes through the gut and through the our vascular system, then comes through blood-brain barrier into the extracellular space. From there, it comes to the essential neuron or essential terminals. From there, how it gets converted into your dopamine and packed into vesicle, and when how they release into the again extracellular space based on the activation of essential neuron. So this is the whole pharmacokinetic or PK/PD model of levodopa. This was integrated into the whole model. So this is the whole model. So the levodopa gut, bloodstream, blood-brain barrier, and it comes to SNC. And again, what happens to the whole modeling, how the arm is trying to reach the top.
Can I ask a quick question?
Yeah.
So this is all in MATLAB and
Yeah,
Symbiology, right? Okay.
Yeah, no, not in biology. It was
Just MATLAB, straight MATLAB. Okay.
Proper coding.
Okay. Yeah. Thanks.
Yeah. And this is the
I have a question. Can I ask a question?
Yes.
In your previous slides, you kind of assumed that the deficit in motor behavior is kind of proportional to the loss of dopaminergic neurons, going from 25% to 75%.
Yes.
But in this type of slides, you see kind of an abrupt going down between 50 and 75%. So it's not like proportional anymore. Do you have any idea what causes this type of, let's say, cliff behavior between 50 and 75% dopaminergic? Because it's in line with the clinical observations. I mean, people start to see symptoms when their loss in dopamine is like 50 to 70%. So any underlying mechanism of the interpretation of this type of sudden decline in performance between 50 and 75%?
You can see there is a sudden increase. If you see 25%, and if you see 50% sudden increase, there is a sudden increase in the behavior in terms of movement time. When there is loss at 50% and 70%, compared to if say 5%, 25%. When able to run the intermediate points, because at that point we didn't have much computational resources to run for the longer cell loss. So as of now, we seen this type of behavior.
But like in part C and D, you see a kind of a it's a nonlinear dose response.
Yeah, it's a nonlinear process. Exactly, it's a nonlinear process. But here at this movement types, it seems like some way linear, but we have to, because there's certain complexity in the model which we haven't considered. Example, the influence of hyperdirect pathway, how that is affecting the whole behavior. And in terms of other neuromodulators in the basal ganglia circuit, example, acetylcholine. These all things we haven't considered in the model. So that might result in the non-linearity.
Yeah. Well, the reason that I ask is because, I mean, it might be of interest for patients to actually have a kind of test in which they can see a very low amount of dopaminergic cell loss, because then you have still time to address the symptom, because usually you're diagnosed when you have already like 70% loss of neurons. So if you can find a biomarker or something like a motor test that allows you to identify maybe a 30% dopaminergic loss, that would be a game changer for those patients.
Yes, that will be very useful. Yes, true.
Okay.
So,
Can I ask like a tag-on question, or go ahead?
Yes.
Yeah. Just a panel A, is there like a cap at the 75% SNC cell loss where the PD2 doesn't, doesn't further change their movement or increase rather their movement time there?
Which one you're talking? Which diagram?
Panel A on the right side, the movement time
Okay.
for the
So yeah, the
75.
75%, why
Yeah,
we clamped at this? Because
yeah.
in the agent, what we clamped is agent, if we are not reached the target within 5 seconds, we are stopping the simulation.
Okay.
So at 70% cell loss, not even one time in 100 trials, the agent was able to reach the target. So that's why there is no standard deviation there.
So for that group, they're failing completely
Yeah, they're
the
failing all the
task.
time.
Okay.
Yeah,
Yeah.
all the time.
Mhm. And that corresponds with the peak velocity being below like
Yeah,
1, well
1.
below 1. Yeah.
It's around 0.5, yes. And you can see average velocity also is very low. It's almost less than 0.1.
Might be interesting to look at like very smaller increments between the 50 and the 75% to see like what Hugo said, if maybe, I know it's not 30%, but like maybe there's some turning point there that might
Yes.
give you some information about the biology.
So yeah, as of now, the simulation when we ran, it was only 8x8 matrix.
Okay.
So only 64 neurons. So at that point, we thought this was the proportion will be useful. If you go with more lower, then again, in terms of computational resources, we have not much at that time to simulate it. But now I think we can do it. But yeah, it's possible.
Cool.
We can increase the number of neurons in SNC and also in the whole model and see how this affects the behavior.
Exactly. Yeah.
Amount of times. Yes, that can be done. Yeah.
Thank you.
Okay. Yes. So yeah, this is the broader like symptoms of the Parkinson, and this is the PKPD model of Levodopa, and here comes the effect of Levodopa. So here we consider another intermediate steps, which is 37 and 62. So which is 25% cell loss, 37% cell loss, 50% cell loss, 62%, 70%. So left set of column is when the projections to only striatum are affected. And the right one is both striatum and subthalamic nucleus get affected. And this is the simulation of the whole model at our time scales. So we ran for 12 hours time scale. And at 2 hours in the simulation, we are giving the levodopa drug. So the first one is for 25%, 50, 37, 50, 62, and 75. You can see when you given the levodopa for the at 25, there's not much. So the green, orange, and blue are the again movement time. So when the blue curve, which is the curve which is running across the horizontally, if that particular curve is in the green, then the particular agent is able to reach the target within 2.5 milliseconds. If it goes beyond 2.5, it is slow movements. And if you go between 4 and 5 seconds, that is very slow movements. So we consider that a normal person can reach the target within 2.5 seconds because it's just 0.8 meters away from the initial position. So we thought that 2.5 seconds is enough to reach the targets. So any individual which reaches the target are in the green. We can see at 25% cell loss, or during all those 12 hours, the agent is able to reach the target. At 37% cell loss, you can see when there is no levodopa, the movements are very slow. Not very slow, which is in the orange. So, and immediately when you give levodopa, the reaching time reduces. It comes to the green region, and has a effect of levodopa wears off. You can see again the curve is going towards the orange. And similarly for 50.
Sorry, can I interrupt? Can you remember us how are you simulating the effect of levodopa in the model?
Yes. This is the effect of the at 2 hours time scale, we are giving the levodopa drug. So first 2 hours, we are just simulating for different cell loss. So you can see for 25% it's in green, for 37 it's going to orange, same 50 orange, 62 it's still in orange, and also 75 still in orange. But as the percentage of cell loss increases, the effect of levodopa decreases. You can see at 70% loss, you can see the model is still in the orange region compared to 62, 50, and 37. Same in the case of when if affecting both the striatum and the subthalamic nucleus. You can see at 2 hours we are giving the levodopa. You can see the effect. The curve is going to green, and as the percentage of cell loss increases, the curve is coming to the orange.
Maybe I missed this, but what's the kind of the standard error in some kind of virtual patient, and how do you define those parameters for the variability?
So the standard error is across multiple trials. So we run this for multiple simulations, like 100 trials, and we'll take the average. Average, the curve is the average line, and the standard deviation is the across the 100 trials.
So it's the same patient that is actually tested over different.
But the initial conditions are different.
Yeah, going from
randomly,
they're different. But I mean, within one line, so it's the same patient that has like 25% cell loss, and then you do 10, 15 tests of the motor behavior.
Again, we don't know if we consider patient, we don't know what percent of cell loss is there in that particular patient, right? That we can only get from imaging. But as of now, we are just assuming that this person cell loss is what is happening. But each trial, there are multiple initial conditions. Example, initial conditions at the case of SNC, there are almost 148 differential equations, and each differential equation starts with different initial condition when you run for every time. So in the case of the whole cortical loop also, there are different initial conditions. And when the arm is also within a standard deviation, within a Gaussian, the arm starts in that Gaussian. So there is always some randomness in the model.
Okay, yeah, thanks.
So it's in a way it's simulating different patients. That's what, again, that's an assumption. With this much randomness and this much noise in the model, it will be able to simulate different patients. So, yeah. And this is again for both the conditions. So this was the comparison based on the literature. So this was the only model which can show both behavior, the effect of dopamine release, and also medication in the single model. This was as of 2022, but if there is a new model which can do all of this, I'm not aware of as of now. And the why, so after doing this, what was the whole idea of developing such models? So the idea was, if we are able to map the patient behavior onto a model and using their hyperparameters, can we profile the patients? That's the whole idea. And using this, coming up with the profiling, and if we can modify this meta parameters, hyperparameters, can we give a targeted therapy, and can we see that improvement in the model, and that type of therapy can we give back to the patient? That was the whole idea about developing this model. And here we started with the reaching task, and that's what we are able to simulate. We're able to get the model hyperparameters. Here in this case, the two hyperparameters we are looking was the percentage of cell loss and what type of projections are lost. So based on that, we tried to classify two different patients here. So one is here is the on the left, on x-axis on the left side is the average movement time, which is coming from the model, and the right-hand side is the UPDRS score for the particular patients. And you can see based on the percentage of cell loss and also the dosage of levodopa, we're able to try to fit the model to the particular patient. So in the case of patient 1, experimental group 2, we're able to see that model 1, PD1, where only projections to striatum is lost, and with the dosage of 150 mg levodopa and 60% cell loss, we can see the model is able to fit the curve. Similarly, in the case of experimental group 1, where the model 2, where the projections to both striatum and subthalamic nucleus are getting affected, I can see that model is able to fit with same dosage of 50 mg levodopa and 62% cell loss. Why we took 150 mg? Because that's what is reported in the literature in this particular experiment, a particular clinical study, and we use almost similar to that dosage. But only another parameter which we worked was on the percentage of cell loss and also the what projections are being lost. So based on that, we're able to fit this model. So based on that, we can profile the patients and give a targeted therapy. So other than the levodopa, the one other targeted therapy which used prominently in Parkinson's disease is the deep brain stimulation. So we want to give simulation to this model, there is no spiking as such, because only spiking neurons in this present model is the SNC. So, and most of the pathological oscillation which you see in the case of Parkinson's disease comes from this subthalamic nucleus and GPE, which is globus pallidus interna. So there is pathological oscillations happening in this. So to develop this pathological oscillation, we replace the rate-coded or not rate-coded Hop oscillators in the present model with the spiking neuron models, which is the easy-rich neurons of subthalamic nucleus and GP neurons. So this is the model. Again, similar model. Only thing is the ST and GP, we are replacing it instead of Hop oscillators, we are replacing it with the spiking neuron models, which is the easy-rich neurons, where we can fine-tune the certain set of parameters in easy-rich neuron model, where it can replicate the firing patterns of different neurons. So here we fine-tune the models for STN and GPE, which can replicate the firing patterns of these particular neurons. And we also model the effect of dopamine on these oscillations. So here, as the dopamine levels increases, the mean firing rate in the case of STN decreases and the case of GP increases. That's what you see in the literature. And also in the case of synchrony in the network, when there's at low dopamine, there is high synchrony. At high dopamine, there's low synchrony. So that's what we see in the case of Parkinson, where due to the loss of dopaminergic tone onto the basal ganglia leads to pathological oscillation, which happens when there is a synchrony in the network. So that's what we are able to see. That's what we are able to formulate it here, where at low dopamine, there'll be high synchrony. At high dopamine, there'll be low synchrony. And when you try to simulate the model, this is what we see in the case of healthy controls. This is the firing pattern of STN. This is GP. This is the network activity. And here is the synchrony in STN and GP. In the case of Parkinson's disease, when there is a loss of dopamine, you can see a bursting type of activity in STN. Same thing we see in GPE. And network activity, you can see there is more synchronous activity. That's what you see in the synchrony of the particular STN and GP. So then we done the whole simulation, like reaching the target. So the blue one is the normal condition, healthy controls, where the particular agent is trying to reach the target. You can see it's reaching within 4.4 seconds. But in the case of the black one, which is the bradykinesia, is the slowness of movements. You can see it's taking longer time. In the case of tremor, you can see it's going doing oscillations. And in the case of rigidity, because of stiffness of muscle activity, you can see there is a distance from the target is constant. That's what you see in the case of movement velocity. You can see in the case of the blue one, which is the control, where you see a high bell curve, again inverted bell curve, where you see lower speed, in the middle higher, and when about to reach the target, the velocity is lower. But in the case of bradykinesis, you can see the velocity going up and down. In the case of tremor also is going up and down. In the case of rigidity, you can see it is the small curve here, which having very low velocity. So the model is able to, with the same spiking network model, they were able to replicate similar results. Then we give the DBS, which is deep brain stimulation, to this particular SN and GP, because these are the prominent regions which are the targets for deep brain stimulation.
Can I ask a quick question?
Yeah.
That synchronous activity in the kind of 10 hertz range in the STN and GPE, is that generated by the STN and GPE themselves, or does that require the full loop through cortex?
This one, you're talking this one?
Yes.
Synchrony. Synchrony is like after the simulation is run, we'll check the activity at each time point. If how many neurons are firing in that particular window or that particular bin. Based on that, we'll calculate the synchrony in the network. Mm-hmm. So in the case of healthy, you can see all are firing at different time points. That's why you see lower synchrony. So when this curve is towards 0, it's low synchrony. When it's towards 1, it's high synchrony, because high synchrony means all neurons are firing together. So that's why you see these oscillations here. There is low synchrony, higher synchrony based on this.
I'm wondering what is causing the higher synchrony.
The higher synchrony is causing due to the increase in. So when you see how you simulate the PD condition in the case of in this model is it reduces the dopaminergic tone onto the striatum. So when the dopaminergic tone is reduces, you can see that's what we formulated. When the dopamine is low, the synchrony in the network is high. When the dopamine is high, the synchrony in the network is low.
So can I ask a quick question? So the dopamine is, how is it exerting that, or is it just like a empirically defined when
So
you
here,
change the
dopamine affecting the, in the case of STN and in the case of GP, it's affecting the lateral connections. So when the dopamine is high, the lateral strength is low. When dopamine is low, the lateral strength is high. That leads to synchrony in the network.
I see. So by changing
So it's the
dopamine,
function of, the function of
you're-
lateral connections.
you're directly changing the firing rate in that
Yes.
sense. Yeah, it's not. Okay.
So that's what you see in the literature as well. So when you see the low dopamine, there is a lot of synchrony in this both nuclei. So that's what we try to formulate. When there's low dopamine, we'll try to affect the lateral connections.
Yep.
Any other questions, guys? I'll just move forward. So here, when we give differential stimulation, so the pink one is the healthy control. Yeah.
I just have one more question. So going back to, sorry, the other slide, the waveforms seem to be distinctly changing. So those aren't changing as a function of dopamine. You're just changing them if the dopamine's high or low.
Yeah. This is changing based on the strength of the lateral connections.
Okay. Yep.
So,
all right.
So it's a network phenomenon. We're just modulating just one parameter in the network, and which results in this type of network behavior.
Okay. Thanks. Sorry. Go ahead.
And sorry, that parameter, is it switching from kind of an irregular or regular spiking to a bursting?
Yes.
Yeah.
Yes.
So
Yes. That's what you see in the
Izikvich neuron. Yeah.
Yeah, Izikvich neurons. Yes.
Mhm.
So these neurons are tuned. There are certain 4 parameters in the model where we can tune the firing pattern based on the experimental data. So using this tuning of this parameter, we simulated the that particular Izikvich neuron model, which is simulate all firing patterns of STL. So here we can show the behavior of single spikes and also bursting behavior.
So how did you get to those tuning values? Did you just kind of
we need to
pick
optimize
what
it.
matched the
Yeah.
literature, basically? Yeah. Okay.
Yeah, from the literature, get the firing patterns, and you can tune these 4 parameters, and you can fit the model.
Okay. Yeah.
You can you can do manually, or you can use the optimization algorithms.
Mhm.
Yes. Yeah. So here, because now we replace the oscillators with the spike network models, then we can now give differential stimulation to this more targeted regions of Parkinson disease. So mostly they target STN, GP, and thalamus. So we haven't done any spiking, replace any rate-coded models in thalamus with the spiking, but we replace the STN and GP. So here we are trying to give deep stimulus to the subthalamic nucleus and see how it is affecting the behavioral patterns. So here is the LFP, which is local field potential of STN across different conditions. So the pink one is the healthy control, where there's no dopaminergic loss. The green one is the Parkinsonian condition, where there is a dopaminergic loss. And the blue one is the STN activity when we give it deep brain stimulation. So you can see it's coming towards the healthy control. And this is what you see when you vary the frequency of deep brain stimulations. And in the case of PD, you can see the peak in the beta frequency. And as you increase the stimulation, you can see the peak is reducing. You can see in the right-hand side that 40 hertz, 60 hertz, 80 hertz, the peak velocity is increasing till 120 hertz. After 120 hertz, it's reducing. The frequency, the peak to peak velocity to reach the target is reducing. So it is known in the literature that when the deep brain stimulation given from the range of 90 to 140, you see the optimum therapeutic effect. So that's what the model was also able to show in the terms of reaching task where the peak velocity is high at the 120 hertz deep brain stimulation. Any doubts till now? So the next part of the work will be to see the effect of both levodopa and also deep brain stimulation together. How the combined effect will be on the this particular reaching task. So that we haven't done yet. We are planning to do, but as of now, this is what we are at. And next is the cognitive loops. So other than the sensory motor loops, we're also looking at the cognitive loops, how basal ganglia involved in different cognitive functions, and can, using the same model architecture, can we replicate certain behaviors. So again, we take the action selection, goal direct behavior, working memory, exploratory behavior, and reward-based learning. So this is the work done in collaboration with University of California, San Diego, where they have the particular subject has performed different cognitive tasks, which is selective attention, responsive inhibition, interference processing, emotional interference processing, all this particular task. And this is the right-hand side is the same model architecture which we used previously, and see if the model is able to perform the task. I'm not going to results of this particular work. You can refer to the links in the below. And another task is the working memory task, which is again use the same model architecture and perform. Here we replace the neurons in the striatum with the flip-flop neurons. Flip-flop is nothing but based on, there are three inputs to the neuron. Based on different inputs, it can store some information or it can remove the information, reset itself. So based on this activity, we will see that there is some working memory capacity at the striatum level, which perform different working memory tasks. So this work also has been done. You can refer to this particular paper. Now, apart from the cognitive loops, we also want to understand how the cerebellum is playing a role. Because when you give deep brain stimulation to Parkinsonian patients, one of the symptoms is there is a lot of balance issue and also the postural issue. So we all know that cerebellum plays a very important role in this particular both functions. So we want to see when deep brain stimulation given to the model, how is cerebellum is responding. So for that, we want to integrate the cerebellum into the whole model architecture and see how this particular behavior will be changing. So this is again future work. We haven't, again, it's a preliminary studies, preliminary results we have, but they're not yet promising results from this work. Yeah. Yes, that's it from my side. If you have any question, I'm happy to answer.
Yeah, well, very, very comprehensive model. Congratulations. I think to try to combine a lot of different brain regions and a lot of different readouts there. I have a question about Parkinson's disease and long-term L-DOPA treatment. So people, of course, having on L-DOPA for quite some time developed L-DOPA-induced dyskinesia. Is your model also able to capture the dyskinesia aspect as a consequence of long-term?
For dyskinesia, one of the main important thing for dyskinesia is to have the serotonin terminals in the striatum. What happens in most of the dyskinesia cases is the dopamine is taken up by these serotonin terminals, and they release irrespective of the activation of dopaminergic neurons. So that's why you see again involuntary movements in the case of levodopa-induced dyskinesias. So in this model, we don't have the serotonin terminals. If we introduce the serotonin terminals or serotonin neurons in the model, we're able to maybe replicate the levodopa-induced dyskinesias.
Okay, yeah. Because LID is usually treated with amantadine, which is a kind of NMDA antagonist. So that would be a good test case for your model to show if NMDA block in the whole system might actually improve the symptoms of L-DOPA-induced dyskinesia. Yeah, thanks.
Yeah, we started with what we know for sure, so that next we can introduce complexity. But other than we only started with what we know for sure. This is what is happening, and we try to incorporate those things. And now we'll incorporate all these different things, adding serotonin neurons and adding the hyperdirect pathway, what happens, and also including the cerebellum, what will happen to the whole sensory motor loops, because that plays a very important role in correction. Again, both basal ganglia and cerebellum plays role in correcting the movements. So what is the role of these two in this? That's also plays an important role. And because it's a spatial thing, we also want in future to introduce hippocampus also and see how that will affect when you do a three-dimensional movement. Instead of right now, it's just in 2D space. If you want to do a three-dimensional movement, how that time the hippocampus plays an important role.
Mhm.
Yeah, thanks.
Yeah. Any other questions, guys?
So you're going to add the hippocampus to the model?
Yeah.
Oh,
As of now, the
cool.
idea is to just introduce the cerebellum. Then in future, we are thinking of adding it.
That's cool.
Because
Yeah.
what we assume is that when there is a goal-directed behavior, initially there's more of basal ganglia and hippocampus, both are involved.
Hmm.
And later, after you learn the task, more of this role of this, both the subcortical regions are reduced. More of now, only the outer loops or the where the information of this task is stored, those only will be activated. The role of basal ganglia and the hippocampus will be reduced. And also in literature, people have seen the basal ganglia also plays important role in spatial navigation along with the hippocampus. So again, because that's why we took the reaching task, which is in a space, so that in future, if we want to include all these things, we have the same task which can do the whole thing. So that's why we started with the reaching task.
Yeah, that's interesting because a lot of treatments focus on the motor deficit, but there's also cognitive decline too.
Exactly.
So, yeah.
Exactly.
Yeah, I was sorry. Yeah.
I was just
I'm-
saying thank you. That's all. Go ahead.
Yeah, this is really great work and very comprehensive. But I am wondering if you've considered including additional ion channels, what role you think that might have. I noticed, I think, an absence of the ATP-dependent potassium channel, which has been considered a potential target in linking mitochondrial dysfunction to altered dopamine release and spiking activity. Have you thought about that?
Yeah. So another parallel which right now we are working on is introducing the ATP dependent potassium channel in the model, because that plays a very important role in multiple ways. One is coming to my PhD hypothesis, which is the energy deficiency might be a reason for this essential cell loss. Introducing this ion channel opens up many more things like this, again studying the hypothesis, and also in the case of protein aggregation. This particular ion channel plays a very important role. I think you presented last time, right, in March. So how this, including this ion channel, will affect the firing patterns. Yes. So you got some good results, right, in this?
Yeah, I was able to fit the in vitro data better with that.
That's nice. Yeah, because we are also looking into that. I think we can talk about this some other time, yes, to
Yeah,
understand.
that's great.
Yeah.
Yeah, it would be great to do a panel where we just compare and contrast the different Parkinson's disease models and the levels of modeling in each of them and the advantages of each of them. And that might be something we do towards the end of the year too. That would be good.
Sure.
Yeah.
So one last question, I think. Is there a model also, are you interested in developing a model also for Huntington disease, which is a kind of somewhat different pathology in which the D2 positive MSN neurons tend to be pathologically deficient?
I can.
So
I didn't get the question.
Are you planning to expand the model to simulate Huntington disease?
Yeah,
Which
that
is
can.
a different pathology, mostly located in the medium spinal neurons of the indirect pathway.
Again, we haven't gone into Huntington yet. Yeah, that is a possibility. You can always do that. You see how that's going to affect, because more of indirect pathway gets activated in that, affected in that particular condition.
And how do you see the jittery movement in your readout there?
Yeah, exactly. We need to see that. Yeah, that will be interesting to see. Without loss of dopamine, how would this going to affect? Yeah, that will be very interesting. And also in future, we also want to introduce the transcriptomic or the gene expression data, so that again we can simulate the Huntington based on the percentage of length of the CAG. Can we see the change in firing pattern that in turn affect the whole network behavior? So that is the intention, because why at 34 length of CAG is you see the symptoms? Why not at 28? Why not at 20? Why at 34 or beyond 34? So why is that the firing pattern? That will be interesting to study. And for that, again, we need to model the all subcellular pathways. How this particular gene is going to affect it. What will the huntingtin protein going to affect the firing patterns? Everything we need to see.
Do we have an hypothesis on how the huntingtin protein actually affects the neuronal firing?
So one of the ideas, again, it's affect the one of the hypotheses we are looking at is the affecting the synaptic vesicle release. So the protein is goes and bind to the synaptic protein complexes, which in turn affect the release of this particular neurotransmitter into the cells. But why are these cells dying in the first place is again a question mark. Because it's high, maybe again protein aggregation might be a reason, but we never know what it is. Is the protein aggregation cause or an effect? That's again, I'm looking into, in the case again Parkinsonian disease, to see the effect. Is it the protein aggregation effect or the cause? So again, linking to Jonas' work where he's trying to understand how this protein aggregation affect the firing patterns and in turn affect the network activity. That's also we are looking into. If we understand in one disease, surely we can easily extrapolate into the other diseases.
Yeah, the recent results from the gene therapy study with uniQure suggest that it's a toxic gain of function that you can reduce by antisense oligos. So I don't know if that could be helpful for identifying the mode of action of pathological Huntington.
Yeah, that can be done. Yes.
Okay, thanks.
Thank you so much.
All right, this was great. I see we're at over time, so maybe we can close the session for today.
Sure.
I want to thank Dr. Vignayanandam Muddapu again for joining us today, and and thank you to everyone else who joined us as well. And we will be we've recorded this session, so hopefully we can put it online, and then we can share it with anyone else who wasn't able to join today. And then maybe we can follow up with a panel later this year, and much more to come. So,
Sure. Thank you so much.
Yeah.
Thank you, guys.
Thank you.